Hello, lightning biologists. Today we're going to do a quick review on DNA replication. And the first thing that we need to remember is that in DNA, it is a double-stranded molecule. And as a double-stranded molecule, the reason that Watson and Crick were able to actually figure out the structure of the molecule was because of the complementary base pairing rule that Chargaff came up with. And so if you recall back, when we have a molecule of DNA, DNA is made up of small units called monomers. And those monomers of DNA are nucleotides. So a nucleotide is made up of three things. It has a phosphate. That phosphate is attached to a 5-carbon sugar, so I draw it as 5 angles of a house. In DNA, that 5-carbon sugar would be deoxyribose. And so this is your sugar. And then on the other end of the sugar, it is attached to a nitrogen base. And in DNA, this is going to be one of 4 nitrogen bases. The nitrogen bases are adenine, and if you have adenine on one strand of your DNA molecule, what would be on the opposite or complementary strand? Well, apples grow on trees. It's one way to remember that adenine always pairs with thymine, and so A and T go together. In the event you have a cytosine on one strand of your DNA, well, a lot of people like to park their cars in a garage. And so this is one way we can remember that. Cytosine always pairs with guanine. You can also think of it as maybe it's important for you to close the gate when you come in and out of your yard. And so base pair ruling is really important, and if you can memorize the base pair ruling, A's go with T's, C's go with G's, you pretty much have the basis of understanding how do I replicate DNA. So when a cell is going through mitosis and it hits that S phase and it has to make a copy of its DNA, how does it do that? Well, it does that because in one strand of DNA, you can see over here, you have a sequence of nitrogen bases, and every time the base pair ruling works. And so if there is a C on the anti-parallel strand, it will be a G. If there's a T, you'll have an A, and so on and so forth. And so that helps us to understand how DNA replication works. So let's just review real quick. Go ahead in your mind. You guys can do this kind of as I go along. If I have on my 3', we'll say this is 3' to 5', and so over here we would be reading it 5' to 3'. If there is an adenine, what would be its complement? Apples grow on trees, and so adenine will always bond with thymine, so thymine would go there. If I have a thymine, what's always going to be the complement of thymine? Well, on trees, you're going to find apples, and so you have adenine. Guanine, what always bonds with guanine is cytosine, and when you have cytosine, guanine will always be its complement. Right here I have another thymine, so I'm going to find its complement adenine, and here I have cytosine, and so its complement is going to be guanine. A and T, C and G. You just have to memorize that in order to always be able to give me the new strand. And so here I have a strand of DNA. So this is the original strand of DNA. We'd say that, it's a double helix. And so right here, I have to unzip using that enzyme helicase. And so as this strand becomes unzipped, you can maybe imagine that we're unzipping it. Right here would be our replication fork. helicase has unzipped it. If you look at the original strand up here, and let's just go with the first few, we would pretend like right here would be the A, the T, the C, the C, the G, the A, and so on. I got that information from right there. I just copied it over here. Well, on the other strand, you would have the T, the A, the G, the G, the C, and the T. And that came from the complementary strand right below. And so you can see we've unzipped. You have to unzip the DNA. And the molecule helicase is going to unzip the helical pattern. Once it's unzipped, you now have the two strands far apart from each other. And so all you have to do now is act like DNA polymerase. DNA polymerase will literally come in and it will read. So right here I have, so I'm going to start right here, okay? So I have an A. DNA polymerase is going to call in a nucleotide that has the complement to A, and so it would call in T. And so T would get called in. Then it's going to move to the next strand, or the next base, which would be T, and it's going to call in an A. It'll move to the next one. There's a C, and so it's going to call in, it's going to call in a G. And you just continue to read down the strand. Now, if this is 3', it's going to read in that direction, but down here, this would be 5'. And so, not that you really have to worry about this or read it in the opposite for the sake of writing down the complement. However, just so you understand that in a cell, it actually would be reading it in this direction, 3' to 5'. And so you see, if we start down here with, I'll erase this real quick, we'll start down here with the A. And so DNA polymerase would call in its complement, which would be a T. Move to the next one, there's a T. DNA polymerase is going to call in an A. Continue to move down, there's a G. So DNA polymerase will call in a C. And it will continue to read the strand from 3' to 5' until it's completely filled in. And once that strand is completely filled in, you're going to notice that you're going to have one strand that will be identical to the original, and you will have a second strand that will be identical to the original. Notice that when DNA replicates, half of the original strand makes up half of each of the new replicated strands of DNA. Okay, so now if you would like to practice and show me that you really are understanding DNA replication, I have posted a Google form that's called DNA Replication Review. That would be awesome if you would fill that in. I would love to hear from you guys and be able to give you some more direct feedback. Now we're going to go on to reading that strand of DNA and actually turning it into RNA and eventually making it into, or using it to make a protein as the cell does.